Now I am going to discuss some problem on stoichiometry of the bioprocess. Now if you look at this problem, this is the Klebsiella aerogenes is produced from glycerol in aerobic culture with ammonia as nitrogen source. Biomass content 8% of ash, 0.4 gram biomass is produced each gram of glycerol consumed, and no major metabolic products are formed. What is the oxygen requirement for this culture in mass term? Now this problem actually deals with you know that single cell protein. Single cell protein is basically the microbial protein, and this is produced by different ways. We can use the carbohydrate for the production of cell mass. We can use the hydrocarbons also for producing cell mass. We can use the alcohol like methyl alcohol, ethyl alcohol for the production of cell mass. Now in this problem we use the Klebsiella, that is Klebsiella aerogenes, that is used the glycerol. Glycerol is a byproduct of the biodiesel producing industry, because when biodiesel is produced from lipid, the byproduct is the glycerol. Now this glycerol they use for the production of this biomass. Now we shall have to find out that this is the aerobic fermentation process, and we shall have to find out that how much oxygen is required for this production process, and yield is 0.4 gram biomass per gram of glycerol consumed. This is the and this basically this is nothing but y x by s. Y x by s is 0.4 gram biomass per gram of glycerol, that is given there. Now glycerol has that empirical formula C3H8O3, and this is equal to, this molecular weight is about 92. Now if you consider the 1 gram carbon atom, then 8 by 3 by O, this is equal to 92 by 3. This is like this. Now empirical formula of the Klebsiella pneumonia, that is assumed to, this is CH 1.7 O 0.43, and then we assume it like this, and actually the molecular weight is coming 23.997, but it contains 8% ash, but that is why we divide by 0.992 to get the whole mass, that how much ash-free mass how much is there, that is we can 26.1 that we get the molecular weight of the biomass, we get it like this. The degree of reduction of substrate we can easily calculate. We have always substrate is glycerol, so we can easily find out the degree of reduction. This is coming about 4.7, and degree of reduction for the biomass I have shown you before, that also calculated at 4.15, and then we calculate the y x by s. This is the gram of cells, gram of cell produced per gram of substrate divided by their respective molecular weight. We get gram of atoms of the cell produced per gram atom of substrate consumed. Now this is equal to yc, and this is coming about 0.4 into multiplied 0.47. Now if you then we calculate this oxygen requirement per 1 carbon atom substrate is coming about 0.687 mole, and if you then what is the total amount of oxygen required per gram of substrate, how we can calculate this is so actually we are getting 0.687 mole of O2 per gram atom substrate. Am I right? So now, what is the molecular weight of that oxygen? That is 0.687 into 32. Am I right? And what is the gram atom of substrate? That this is gram of oxygen, and gram atom of substrate is how much? That is 1 gram atom of substrate is about 92 divided by 3. So if you, this is the gram of substrate. The molecular weight of 1 gram atom of substrate is 92 by 3. So if you divide by this, it is coming about 0.72 gram of oxygen per gram of substrate. So this you can easily calculate. This is not a problem for us. Now next problem, that again this is with the Candida utilis. Following the macroscopic coefficient should be determined for the cultivation of the yeast. Candida utilis can utilize both hexose and pentose sugar on ethanol. Substrate consumption for biomass production, oxygen consumption, and fermentation, heat produced, and determination stoichiometry of the process. So these are the different parameters you have to determine, and as I pointed out before, that whenever we want to solve any kind of stoichiometry problem, first we should have some idea about the process, otherwise we cannot solve this problem. Now here Candida utilis is considered as a fodder yeast. Fodder yeast means this is used as a animal feed. Now and the speciality of this yeast is that it can use both the hexose and pentose sugar. Now here this Candida utilis is utilizing the ethanol as a substrate for the production of the biomass. So we what we required basically, that we required for cell mass formation, we required carbonaceous substrate, but plus nitrogen source. Nitrogen also contribute for the cell mass formation. And I told you the aerobic process is required. Why aerobic process? Because the cell mass growth in the aerobic process is 10 times faster as compared to that of anaerobic fermentation process. So we required oxygen for. So that is why in case of this stoichiometric equation, when you write, when you find that this is ethanol, that which is a gram atom of substrate. With the ethanol, when we write it is like this, because ethanol I showed you before that C2H5OH. Now if you write in the empirical formula like C2H6O, and when you write per gram atom carbon, it is like 3O.5. So this is per gram atom carbon. This is exactly we have written. This is ethanol. Then B moles of the oxygen and C moles of ammonia, when react, that in combination they form the D moles of the per gram atom of biomass, then F carbon dioxide and G water. So here this is the substrate, and this is the substrate, and this is the biomass. So we can easily calculate the degree of reduction of substrate and degree of reduction of biomass. And degree of reduction of substrate, how you can calculate here? You see this C stage is 4 and H is 3 and O is 2 into 0.5. This is coming about 6. Similarly, we can calculate for biomass. It is C is 4, H is 1.79, then 2 into 0.5 minus 3 into 0.2. It is coming about 0.19. Now similarly, that sigma S and sigma B we can calculate by with respect to gram carbon. This 12 divided by molecular weight of the substrate and 12 divided by the molecular weight of the biomass. So all are coming. This is 0.522 and this is 0.488. Now when you this, since it is a aerobic process, the thermodynamic coefficient is assumed to be 0.5 to 0.6, and we can assume 0.6 because and we assume there is no other product excepting biomass. So epsilon p can be assumed to be 0. So then eta will be equal to 0.5, and eta if the y x by s, then we can calculate with the help of this degree of reduction, and the you have sigma s. We can easily find out, and we find out that this is coming about 0.92 gram biomass per gram of substrate consumed. And similarly we can calculate the oxygen consumption. 3 eta divided by 2 sigma is gamma b, sigma b gamma b 1 minus eta minus epsilon equal to 0, as no product has been formed other than biomass. Then we can calculate this is coming about 1.1 gram biomass gram of oxygen use. The oxygen demand B, that can be calculated like this, and B is coming around, this is 0.6, this is gram mole of oxygen per gram atom of substrate consumed. So now if a B, if you what is the heat evolved in this reaction is equal to 4 into Q0 into B, that is 4 into 133 into 0.6, that is around 319.2 kilojoule per gram atom substrate. Now per gram atom substrate, if you want to find out that per gram of substrate, then you have to divide by the with respect to that the gram atom molecular weight of the gram atom of substrate, that is 23, and it is coming about 13.8 then kg per gram of substrate. So we can calculate the molecular weight of the substrate is about gram atom substrate is 23. So we can calculate like this. Now if you when you do the stoichiometric equation balance, write the stoichiometric equation, we can easily do the carbon balance. Now if you look at here, this carbon is contributing in 2 different forms. So if you do the carbon balance, this is one carbon, one is equal to D plus F, am I right? D plus F. So we have already calculated the D because the yield coefficient already we calculated. So F equal to 1 minus D. D we already calculated here. D is coming here is this is YC. We have YC is zeta, sigma is by sigma B. It is nothing but the YC is nothing but D. This is equal to 0.859, and we minus this, we will get the F value. We get about 0.14. Similarly, we can do the oxygen balance. Oxygen, we have 3 different forms. We have oxygen here, oxygen here, oxygen here, oxygen here. So all more or less all excepting ammonia, all places we have oxygen. So we can make an oxygen balance. This is 0.5 into B into, you see that how we can P into 2 into B, because 2 then 0.5 into D, then 2 into F and G. Then if you this equation, then we can find out G value, everything is known, then we can find out G equal to this. Similarly, we can do the hydrogen balance also. Hydrogen we find here substrate, ammonia, and this biomass and water molecule. Then we can do this calculation, and we can find out the C value is 0.15. And nitrogen balance is very simple. Nitrogen only contribute for the biomass. So this 0.2 into D equal to C. So you know the value of D, so we can find out the value of C. So if once we know all these things, then we put this value, that different coefficient we can put it here, and we can put this equation, we get the complete stoichiometric equation. Now this why this equation is required, because this gives you the idea of what is the how the different components contribute for the products and other byproduct formation process. So this is very essential for the industry to find out the stoichiometry of this particular process. Now another thing I pointed out that the stoichiometry not only gives us the interrelationship of different molecules present in the reaction mixture, but also gives the idea that on the validity of the experimental results. Now this let us see how we can find out that. Now there is a problem. This is called during the anaerobic microbial production of ethanol on complex media containing sugar, the following experimental data were recorded from the batch culture process. The concentration is gram per liter. This concentration all are in gram per liter. So this is the time of fermentation. So this is a batch process. So batch process basically it looks like this. It is in the batch process it's like this. So what you do, we put here ethanol, put here sugar in the media, and time to time we draw the sample, and we find out at different time what is the cell mass production, what is the cell mass or biomass production. Then we have what is the sugar concentration, and what is the ethanol concentration, ethanol concentration that we have. So we can find out that the different concentration we can find out in this reactor. This is reported here. Now we shall have to find out the data that is reported here, that is right or wrong, and which data you feel that requires some kind of corrections. So check the data reported is correct or not. So let us see how we can do the analysis. Now sugar has the formula of that sugar has the formula is what, that is C12H22O11. Am I right? So so this is the when it hydrolyzes, it produces 2 C6H12O6. This is in presence of water. They produce like this. So so initially this is considered the carbohydrate. This is CH2O. So this is we write this empirical formula. This is in case of sugar is CH2O, ethanol CH3O.5, and biomass is a per gram atom carbon. What is that we find out. Now from the experimental results you see that we know the initial substrate concentration and final substrate concentration, initial product concentration and final product concentration, initial biomass concentration and final biomass concentration, that we know. Now since we know that, we can easily calculate x minus x0. There is biomass, initial biomass concentration, now final biomass concentration and initial biomass concentration, and it is the substrate concentration will be the S0 minus S, that is initial substrate concentration minus final substrate concentration. So this ratio is coming about 0.106. Now P by S is coming at is like this. This is a little bit mistake. This should be S0 minus S, and here it is P minus P0. This is coming about 0.539. Now from this this empirical formula we can find out gamma S and gamma B and gamma P. We can at the same time we can calculate the sigma S, sigma B and sigma P we can calculate, and if you know that, then we can find out y x by S equal to eta sigma S, gamma S, sigma B and gamma P. So we can put this value and find out what is the value of eta, and similarly y P by S we know that then we can find out what is the value of epsilon, Now we know that because this ethanol fermentation usually carry out under anaerobic conditions, and under anaerobic condition we know the thermodynamic efficiency of the process is about 0.7. So now we shall have to check that whether this calculated value, because what we reported here thermodynamic efficiency, this is equal to eta plus epsilon p. Okay. Now what we got, this is about 0.135 plus 0.1055. So 1055. So this is much greater than 0.7, because in the anaerobic process, this is anaerobic process, this thermodynamic efficiency is 0.7. So it is obvious that your results have some whatever data you have reported here, that has some mistakes. So we shall have to find out that where is the mistakes. Now when we try to do that, we find out that which component plays very important role, might be having some error. As for example, that ethanol we usually estimate by gas chromatograph, and gas chromatograph we can find out the ethanol concentration accurately. So there will not be much error. We can expect that the estimation of ethanol doesn't have much of error, and usually the sugar concentration in the lab we usually estimated by the DNS method, and DNS method is actually not for the sugar actual sugar estimation. This is used for finding out the reducing sugar concentration. So maybe due to some experiment error we get this some erroneous result here, and then we try to what we have done, we multiplied the substrate concentration with some other factor like 1.2, 1.5, 1.8 to 1.6, 1.7, different factors we multiplied and try to find out that what factors gives us the eta plus epsilon value 0.7, and we find that when we multiplied by 1.7 substrate concentration, then we get the eta plus epsilon value, epsilon p value is 0.7. That means that whatever data we reported for the mass concentration, that is to be multiplied by 1.7, then we will get the correct result, that is thermodynamics efficiency will be 0.7. This is how we can find out the validity of the experimental results. Now next problem that I want to discuss about the anaerobic digestion process, which is largely in operation in the different places, because you know that anaerobic digestion process we can produce methane and carbon dioxide, and biogas we have largely used in different places, not only in the industry. Nowadays biogas is used for the operation of different bus and different vehicles. So this problem related to that, and we have seen that this problem, the elemental analysis of the refuse, it contains about 75.6% of weight by weight of organic matter, and whose composition is this, containing 24% is ash, and minimum methane produced by the anaerobic digestion is calculated by kg of dry refuse, also determine the stoichiometry of the process, that you shall have to calculate. Now for the again I mentioned that whenever we shall have to do any kind of stoichiometric analysis, first we shall have to know the process. Now anaerobic digestion process, how it produces methane and carbon dioxide, because what is happening that organic residue comprises of lot of polymeric substances. This undergo the hydrolysis. As for example, that glucose as for example cellulose, hemicellulose, starch and protein, all these big molecule, even lipid, they are the bigger molecules. They are with the help of hydrolyzing enzyme like cellulase, amylases, then protease, lipase, they degraded to the smaller molecules. Then it will be becoming it will be solubilized. After that, this undergo the acid with the help of acidogenic reaction. This undergo the metabolic pathway like Embden-Mayer pathway, and then it produces different organic acid like volatile organic acid like acetic acid, propionic acid, butyric acid, and then it is converted to methane and carbon dioxide. So basically that the reaction is that refuse reaction is first undergo hydrolysis, then intermediately it produces organic acid, then produce methane and carbon dioxide, and also it produces ammonia and some kind of hydrogen ion. So this is how we have written this equation. This is the substrate, and then this is the water molecule, then methane, then it produces carbon dioxide and ammonia, and then hydrogen ion. Now here we have so we considered the methane as a product, and we ignored the cell mass produced, because this, since it is the anaerobic fermentation process, we assume the biomass formation here is negligible. So if you do that, then sigma s we can calculate it with respect to this substrate, and with respect to this product we can also calculate the gamma s and gamma p, that we can calculate. Both the things we can calculate. Then we can assume this anaerobic process epsilon p is 0.7, and if it is 0.7, we can easily calculate that how much gram of methane is produced per gram of substrate consumed, because y p by s is the yield coefficient. Yield coefficient is the gram of product, that is gram of methane produced per gram of substrate consumed. So this is coming about 0.176, but this is we considered the per kg of organic biomass, but organic biomass contained about 75%, the total organic residue containing the 75% organic biomass. So you multiply it by 0.76, then you will get 0.34 gram of methane per gram of substrate. So we can easily calculate how much minimum gram of methane can be produced. Then we can also find out this p value, that different this component that we required for this. We can one by one we can calculate. You know this equation, the b is a product, that how much product, that is yc value we can easily calculate. And this is this is yp value. Yp value is we can calculate like this ratio, and once we know this b value, we can find out the c value, because b plus you see that b plus c, the b plus c carbon balance, b plus c equal to 1, and then we can easily calculate the c value. Like this we can find out the d and a value, and when you calculate all this balance like before, we put this coefficient here in this equation, and we get the complete stoichiometric equation. So this is how we can write the complete stoichiometric equation. So this is very much required for all the fermentation process. I hope this will be very useful for doing the material analysis of the process. Thank you.